When we talk about countercurrent mechanism, many question comes to our mind. Why it is called countercurrent mechanism? What is countercurrent mechanism? Where does it occur? And how does it occur? We are talking about the countercurrent mechanism that takes place in the kidney. We have already studied that there are two types of nephron in our kidney. One is cortical nephron and another is juxtamedullary nephron. As you can see in this figure, cortical nephron, they are short nephron and they have very short Henle's loop. On the other hand, juxtamedullary nephron, they have a long Henle's loop. You can see a very long Henle's loop is there in the juxtamedullary nephron. Now, the purpose of this Henle's loop is the concentration of urine. Longer is the Henle's loop, more concentrated urine will be produced. It means cortical nephron are not playing any role in the concentration of urine. Another thing that you can notice is that around the nephron, there is a network of capillaries. Efferent arteriole from the renal artery, a branch of the renal artery, divide into capillaries, form a network of capillary in the Bowman capsule, and this form glomerulus. Now these capillaries, they unite and then they come out as efferent arteriole. This efferent arteriole, it further breaks down into a network of capillary around these Henle's loop, and this is called vasa recta. Vasa recta and Henle's loop, these form counter current mechanism along with the collecting duct. So there are three things. One is Henle's loop, second is vasa recta, and third is collecting duct. So these three are components of counter current mechanism. Now let us study this. First question. What is counter current mechanism? Counter means opposite. Current means flow. So there is flow of fluid. Here it is filtrate or urine in the opposite direction, where in the Henle's loop, you can see this is Henle's loop, this is the descending limb, and this is the ascending limb. So you can see in the descending limb, filtrate is going in the downward direction, and in the ascending limb, it is going in the upward direction. So opposite flow. So this constitute the first counter current. Second thing, vasa recta. Vasa recta is also having the descending and the ascending limb. Again, it makes the counter current. 2. Number 3. Where there is descending limb of Henle's loop, there is ascending loop of vasa recta. So this form third counter current. So all these counter current, these are responsible for making the concentrated urine. Now this concentration is basically occurring in the inner medulla. Now let us see how does it occur. When the filtration occurs in the Bowman's capsule cavity, the filtrate is collected here, and this filtrate is having the osmolarity of 300 milliosmol. Our blood is also having the same osmolarity. So you can say this is now isotonic to our blood. When this filtrate comes into PCT, its osmolarity is still 300. Now by the time this is going to reach the tip of the Henle's loop, its osmolarity changes to 1200. So now let us see how does this osmolarity changes. When the filtrate is passing into PCT, it is isotonic. Now when it comes to the descending limb of the Henle's loop, this loop is permeable to water. I am saying this is permeable to water, but this is impermeable to salts. So only water can come out of it. Now the area that is surrounding this nephron, this is called interstitial fluid. So here it is interstitial fluid. So when the water is coming out of this, the interstitial fluid is now having more water, but inside the Henle's loop, it become concentrated because still salt are there. Now as it move down, it become hypertonic, then more hypertonic, and at the tip, it is most hypertonic. Its osmolarity is 1200 milliosmol, almost 4 times of the initial osmolarity. When it is moving up in the ascending limb, now ascending limb, they are impermeable to water, but these are permeable to salt. Now, first of all, this salt will come out by the passive transport process, but later on in the thick limb of the ascending limb, this salt come out by the active transport process. But active or passive, only salt is coming out. So what would happen? Now in the Henle's loop, salt is not there because salt has moved out. So now it becomes hypotonic. So you see, as this move up, its osmolarity decreases. From 1200, it comes down to 100 milliosmol. So it is very dilute now. If there is plenty of fluid in our body, in that condition, this may be released as such and get collected in the urine and goes out. So in that case, we are passing hypotonic urine. I am saying when we are having lots of water fluid in our body, and body has to get rid of that excess water. In that condition, there is no need for reabsorption of water because already water has moved out here. Now what is vasa recta doing here? See here, when this ascending limb is there, there is this descending limb of vasa recta. So NaCl that is coming out from this is directly going inside this. And water is coming out of this. This is near to this. So when water is coming out of this, inside the blood, inside this vasa recta, blood osmolarity is changing. And now at this tip, it become 1200. Look at this. Here it is isotonic to this. Now look at this ascending limb of vasa recta. This ascending limb is near to this. The water that has come out of this is entering into this. And not only this water is entering into this water is also entering into this. Okay, so water is moving out from this limb, but water again is entering into this vasa recta. So now interstitial fluid is not having water. You can, earlier we have said that interstitial fluid is having water here. But now I am telling you that this complete water, or let us do one thing. We draw this ascending limb here. This is the ascending limb here. And this is the descending limb of vasa recta. So what I am saying that this NaCl that has come out is going to enter here. But in turn, water is coming out. This is called counter current exchange. So in this counter current exchange is occurring. One NaCl is going in, one H2O is coming out. In this case, ascending limb, this water is going in, this water is also going in. So now there is no water in the interstitial fluid. Okay? And the NaCl that has come out of it, it enter into this. So NaCl keep on circulating. That it comes out, goes in. Comes out, goes in. Because from here, NaCl is coming out. The NaCl that has come out from the ascending limb goes into descending limb, then again come out in the ascending limb. So what is happening here? There is more concentration of NaCl in this area. And why there is more concentration of NaCl in this area? What is the significance of more solute here? We know that water always move from high concentration to low concentration. So when more solute is there, there will be less water concentration. So water will be reabsorbed more and more. More water will come out from this loop of Henle's and more water will go back into vasa recta. You can check here also that how this countercurrent mechanism is there. I told you that one mechanism is there, one mechanism is this, one is these two loops. In this way, there are so many countercurrent exchange are occurring simultaneously. This I have drawn vasa recta, ascending loop. This is descending limb of Henle's. This is Henle's loop. This is Henle's loop. And this is vasa recta. So this is descending limb of vasa recta. This is ascending limb of vasa recta. And this is Henle's loop, ascending loop. Right? I think now this is clear to you that how many countercurrent we are having. One countercurrent, two countercurrent, three itself this, and fourth itself this. Now what I am telling you that water is coming out from the descending limb, right? But simultaneously this is entering here. This water is entering here. entering here. Okay, so at the tip, osmolarity in the blood is also 1200. Osmolarity here is also 1200. Again, now look at this. I am telling you that only NaCl can come out of it, right? The NaCl that is coming out of this is entering here. But in turn, what is coming out? Water. The NaCl that has entered here will come out from here. Again exchange. And this HCl will come inside this. This water entered into this. So you can see that the water that is given out from this descending limb of Henle's loop, water that comes out from this vasa recta descending limb, they both these water molecules, they are absorbed by the ascending limb of vasa recta. So whole water is reabsorbed. The only thing is recirculated here is NaCl. And what is the purpose of this? Just now I have told you, so that more solute concentration can be there. This is the purpose of circulating this NaCl there. See, look at this. This is the outer cortex area. Okay, so in this, the osmolarity is 300 only. And this is the outer medullary area, and ye wala, this area, the dark purple area, this area is inner medullary area. So this 1200 milliosmol osmolarity is because of the circulation of NaCl. Collecting duct are also helping in this. How collecting ducts are helping in it? A certain amount of the urea also reabsorbed in this area. And this urea, a certain amount of this urea will get reabsorbed back into the ascending limb, and again it will come here. So this urea also keep on circulating. So urea is there, NaCl is there. So because of this urea and NaCl, this inner medullary area is having such a high osmolarity. And because of this high osmolarity in this area, more and more water reabsorption will occur. One thing more, one point you have to note, that 1200 milliosmolarity occurs only when there is a need to pass the concentrated urine. So this occurs under the influence of hormone ADH, antidiuretic hormone. So when we have to produce concentrated urine, then this ADH, they will act on this collecting duct. Otherwise, collecting tubule are not reabsorbing water, but under the influence of ADH, the membrane of collecting tubule, they become permeable to water. And water start moving out of these collecting tubules. And a concentrated urine is passed. So now I hope that the process of countercurrent mechanism that result in the formation of concentrated urine is clear to you.